All right, so moving on to the next section, continuing on, we're going to talk a little bit more about cell structure and function. Again, I know this stuff is like the basics. I get that. It gets more interesting from here, I swear. Right? We just want to make sure everyone's on the same level, so that way when we talk about the more complicated stuff, everyone can kind of hit the ground running with that. So just keep with me for the next section or two. Okay, so here we're going to talk about cell structure and some of the function. We're going to talk about plasma membranes and organelles and all that sort of thing. And then we'll talk a little bit about enzyme function and energy production. We'll get more into detail on that. And anyone have like nightmares of having to memorize like the TCA cycle, right? I'm not going to have you memorize the entire cycle. We're going to memorize just what kind of you need to know from it, right? Again, because the whole point here is not to make you guys chemists, not to make you pharmacists or anything like that. You want to be good practicing PAs. And so that's why we're going to get just the nitty gritty of this stuff down. But we'll talk about more in that detail in the next section. Anywho, into cell structure itself, I'm sure people have seen this a lot. Anyone ever had to like know? I remember when I was like in high school biology, I had to like make a big Play-Doh cell and had to like label everyone. Anyone have to do that? Yeah. So again, here, but you guys are just paying a lot more money for it now, right? Just kidding. Anyway, so again, cells, the basic functional unit within the body here, and again, depending on what type of cell you're dealing with, will dictate what type of tissue you're going to be dealing with, what type of organ it's going to be. Principal parts, so they all share the same principal parts, whether they're dealing with a neuron versus a GI cell. They're going to have a plasma cell membrane. Again, we mentioned that's going to help them maintain that integrity, keep things out that should be out, and keep things in that should be in. The cytoplasm, what is cytoplasm? It's kind of all the goop and stuff that's going to be within the cell itself, right? It's going to provide some of that fluidity there. And then the organelles are going to be the specific organs, essentially, of the cells. They're going to provide some specific function. And then the nucleus is going to be very important here. Now, do all cells have a nucleus in the human body? No. Do all cells just have, those that have a nucleus, do they just have one? Usually, but you're going to find there's some tissue types, some cell types are going to have multiple. So, for instance, yesterday we mentioned the striated skeletal muscle that has multinucleated cells within it. That's going to have multiple nuclei, right? Versus, most cells you're going to find just have one nucleus. But again, things like red blood cells might not have any, right? But what is stored in the nucleus? DNA, right? That's where we're going to get all of our genetic information. Usually, at least some, the cells that do not have a nucleus usually have a precursor that had a nucleus at one point, but once they mature, that's when they end up losing that. Again, you can reference back to this slide here for the individual organelles and what their specific function are going to be. We're going to talk about these as we go forward, but again, you can reference back to this. So again, we mentioned the cell membrane, a lot of good function here, and mainly going to be for structural support. The other big thing I mentioned as well is going to be for regulating transport through the membrane. And we'll talk about things like passive transport. We're going to talk about things like facilitated transport in much greater detail later. But again, some things can cross by themselves very easily. Right, things like water are actually able to cross lipid bilayers with some ease. You're going to find things like certain molecules can go across very easily. In some cases, things are going to be absolutely blocked from going through, and they may need things like transport proteins in order to make that happen. Right. So, for instance, glucose doesn't just go into our cells very passively. We actually need insulin as a factor there to have start the cell signaling process to allow those transporters to let glucose through. Essentially, and that's important. We regulate that very tightly. You're going to find that receptors for things like hormones and other molecules are going to be lodged here within the cell membrane as well. Most drugs you're going to find, if they interact with a receptor, typically are going to be on the cell surface here. They're going to kick off what we call a secondary messenger system, which we'll talk about in pharma later on. But basically, when drugs are interacting with the body, chances are it's going to be at the cell surface, right? The other thing you can find as well are going to be these markers. This is what's going to mark our body as self versus like an antigen. The immune system is going to mark it as attacking, right? So again, your T cells here are going to be one of the important immune cells we'll talk about later. But again, it can mark something as our cells versus something that's foreign and mark it for eventual destruction, right? Which is a big problem when you think about if you were to say take someone's organ and transplant it into a new patient, right? What's one of the things you have to worry about? Rejection, right? Because your body recognizes that as a foreign protein, as foreign cells, we want to get rid of it. And so we have to do things like give them immunosuppressants that prevent the immune system from attacking it inappropriately. So again, we'll talk about that later on. We get into things, topics like transplant and immunosuppressants. But again, it's very important that the cell surfaces have those markers there to kind of identify whether it's our cell or something else. So some of the cell processes that can occur within the cell membrane are going to be things like phagocytosis and pinocytosis. Can anyone kind of describe what those are?
Phagocytosis where it It's a vacuole to bring inside.
Yeah, so the way I always reference it is like phagocytosis is cell eating, so it kind of munches on it, and then pinocytosis is more like cell drinking, essentially, right? So it's going to be for smaller molecules typically, but basically it's going to be able to kind of go in and bind something up and suck it into the cell, right? So again, in a lot of cases, this is not the primary way that certain products will get into the cells, but for things like macrophages, you guys are familiar with macrophages? What do those do? They're like Pac-Man, they go and gobble stuff up that they want to get rid of, right? And so again, phagocytosis is an important process there. Again, good for body defense, inflammation, apoptosis when you're ready to get rid of a cell, right? So it triggers off that cell death. And phagocytosis is going to be very important for that. Again, pinocytosis is good for small molecules. So again, you can imagine the cell producing that podocyte is going to kind of go in and engulf whatever the molecule is, or there's a bacteria or something else. Again, like the Pac-Man reference there. Whoever made this is a very sick individual. That's terrifying. But you can kind of think about that as like a cell kind of eating up various things. So exocytosis, what is that?
Expelling something from the cell.
Yeah, expelling something. This is very important when we were thinking about things like, we talk about like psych meds, and we talk about behavioral health. We mentioned serotonin, like it was a really important neurotransmitter for that. But you're going to find that exocytosis is the main way that things like neurons are going to be able to expel molecules in order to interact with the next neuron in the chain there, right? From the axon to the dendrite, so the next neuron. Exocytosis happens there very frequently. Anytime you're getting rid of waste products and things like that from the cell, this is usually the process going to be happening there as well, right? So we talk about things like lysosomes and things like that. They're eventually going to be undergoing exocytosis to get rid of those products that the cell can't really deal with anymore. Again, just know, and what do you call this term, this vesicle, right? So the vesicle will end up fusing, because again, what's surrounding this vesicle? Yes, basically it's a cell membrane. So still that same phospholipid bilayer is able to kind of sequester whatever's in those within that vacuole, and is able to vesicle and eventually fuse with the cell membrane on the cell surface and get rid of it, essentially, right? So again, this is just a kind of a little pinched off portion of the cell membrane. It can fuse with it very easily, and again, just kind of integrate itself very well. Okay, other cell structures you might find, things like cilia. We mentioned cilia yesterday, especially like within the case of like the lungs. What do the cilia do? Yeah, they're kind of modal. They are going to be kind of, they usually have a kind of a synchronized sort of coordinated effort to kind of move things in a specific direction. I mentioned these also help with cell surface area quite a bit as well. But the lungs are the primary place I think of as ciliated tissue, because again, we want to get that mucus out of the lungs. We want to make sure to get rid of microbes and foreign bodies and things like that out of there. And again, when you have diseases that can modify that, like chronic smoking, definitely damages a lot of those ciliated cells. You end up losing a lot of those, and that can lead to things like pneumonias. Respiratory infections of all types can develop with that. So I have just some other pictures of cilia you might see. So you can do little tiny little hair things. They can kind of flap in a coordinated sort of motion. We also have a flagellum, again, or flagellum common in human cells. No, you normally think like bacteria, like protozoas, things like that have like flagellum. Actually, the sperm is the only cell in the human body that actually has a flagellum. Again, it's made for transport, essentially, or made to motility, essentially, to kind of move itself from one place to the other, right? Next one, the microvilli. What are microvilli good for? Absorption, very good. So again, you're going to find that the majority of your GI tract is going to be lined with these microvilli. Instead of having just kind of a smooth surface for food and things like nutrients to interact with, you have these kind of little feet or little appendages kind of sticking out, and it increases surface area dramatically. And this is where a lot of absorption of nutrients, drugs, absorption of everything happens to the GI tract, these microvilli, right? If you were to lose these, right, say you would have damage done to the tissue, say you have something like, say, a severe inflammatory condition of the bowel. Anyone know of any of those? Yeah, Crohn's disease could be an example. You might actually lose some of that, do that tissue damage over time. And guess what? Now you have malabsorption. You're going to have a harder time absorbing nutrients appropriately because the microvilli might not be there or may not be functioning quite as well. So absorption is really an important portion here, and why the GI tract is so good at absorbing things. Again, just kind of see how things can intersperse themselves within the microvilli, and how that cell surface area kind of goes up pretty significantly. Next, we have our endoplasmic reticulum. There's two varieties of that. We have the rough and the smooth endoplasmic reticulum. What is the rough one covered in? Yeah, ribosomes. What are ribosomes good for? Yeah, it's going to be good for protein production. So if you think ribosomes, think rough endoplasmic reticulum, that's where mainly you're going to have most protein production is going to be happening. We'll get into more detail on that a little bit later, but that's the primary space for it. The agranular or smooth endoplasmic reticulum, anyone we use that for? Yeah, some transport of proteins. Again, it's going to have a little bit more of a specialized sort of function here. One of the big things I think about a lot, especially with skeletal muscle cells, is going to be the sarcoplasmic reticulum, and that's actually very similar to the smooth endoplasmic reticulum here. It's a big source for calcium storage. Calcium is going to be a big cell signaler. You're going to see that when you release calcium within a cell, it's going to cause all sorts of things to happen, and that sarcoplasmic reticulum is a big place for that to occur there. Okay. Okay. Then we have the cytoskeleton. What does this do for us? Yeah, basically provides structure to the cell. Again, you don't want to have a cell that's like completely squishy and can flatten itself out. It needs to maintain some sort of stability there. Because again, the lipid bilayer itself, while it provides kind of a nice coating, it's not really all that structurally sound from a stability standpoint. That's what the cytoskeleton's there for. Notice that you're going to have these also be very important when you're dealing with, again, the cytoskeleton is made up of these microtubules. This is very important during, what's this process here happening? Yeah, it's mitosis or cell division occurring here. So again, this is actually going to be pulling, we'll see this later, they're pulling those chromosomes apart. But again, microtubules are very, very important in forming the cytoskeleton and helping with things like mitosis and cellular division. Here's some other pictures of kind of what the cytoskeleton kind of looks like. You can see how it's kind of projecting out and kind of provides some stability for your cells there. Okay. Next, we have our lysosomes. What do these do for us? Yeah, they're going to be basically going to be digesting things essentially. So if you have, say, certain waste products or certain nutrients have been brought into the cell, lysosomes can be very useful there. They contain very highly reactive chemicals that are able to break down these products. In some cases, you may find that you have things like these kind of primary lysosomes that will be formed that they will only contain the digestive enzymes, right? As you incorporate that with, say, like a food vacuole or some other waste product you're trying to metabolize, you're going to find this, we're going to form a secondary lysosome, and then eventually you're going to develop this residual body. This is the stuff you just want to get rid of, and then it undergoes what? To get it out of the cell. Yeah, the exocytosis. That's where one of those processes there where you can get rid of waste products that the cell just can't use anymore. But lysosomes are the important thing there to make that work. Also, you can see like bacteria, if a bacteria were to bind that up, or to be in a lysosome, that's how we kind of destroy those and things like macrophages. Some more pictures kind of showing how these are going to be working. Notice that these primary lysosomes are going to interact with, say, something that we want to get rid of. It's going to form that secondary lysosome, and then you have that residual body that gets excreted out. Okay, we also have some peroxisomes, kind of in a similar vein to the lysosomes, but these are good for catalyzing certain reactions and causing metabolism to occur. When I say metabolism, what does that mean? Yeah, usually breaking something down essentially, right? So, for instance, when you have a lot of peroxisomes, especially in the liver, and one place, and again, maybe over the long holiday break, not that you guys would ever do this, but some people consume alcohol on holidays. But this is one of the ways we can actually detoxify alcohol within the livers with these peroxisomes. Typically, you think peroxisome, you think hydrogen peroxide, which is H2O2. Is this a highly reactive molecule or pretty stable? Highly reactive. Hydrogen, again, imagine if you have like I'm sure everyone at their parents' house has like a bottle of hydrogen peroxide that's been sitting around since like the 80s. If you open it up, guess what? It's not hydrogen peroxide anymore, it's just water, right? Because again, it breaks down very easily because it's so reactive. But H2O2 is a great molecule to catalyze certain reactions and to start breaking down certain products. And so this is one place you might actually use that. Eventually it breaks down into H2O, just plain water by itself. But that, in the meantime, it forms a very reactive medium that can interact with all sorts of things. Okay, next we have the mitochondria. Again, the mitochondria is powerhouse of the cell, right? We're going to get talking about this when we get into talking about metabolism and aerobic respiration or aerobic metabolism of sugars and things like that later on. But again, this is the main place where what are we producing here? ATP, right? ATP is going to be the main energy currency of the body. It's a very highly energetic molecule. We need that for all sorts of reactions to occur within the body. So just know this is where this is being formed. We're going to talk about this in much greater detail in the next section. We talk about ATP production here. Again, I'm not going to make you memorize all the steps of the TCA cycle and all of that, but we're going to just know basically like what you're putting into the system and kind of what you're getting out of it. And so that's going to be the important things to take away there. Because again, this is useful for aerobic metabolism, but what's the other flip side of that? If you don't have oxygen around, what do? Yeah, anaerobic metabolism. That can actually be a big cause for problems in all sorts of disease states, right? So especially if you think about things like sepsis or you think about things like certain poisonings, if you switch over to anaerobic metabolism, that gets pretty bad. Because anyone know what you produce with anaerobic metabolism? Lactic acid, and that again, what's that going to do to your blood pH? Drop it, right? Because again, it's an acid. It's going to make it more acidic, so that can be a big problem in a lot of these patients. So mitochondria are very important. They have good function. If they don't, then patients don't do too well in the long term. All right, then we have the cell nucleus. You mentioned the nucleus is where? It's where all the genetic information is, where the DNA lives, essentially. Again, not every cell is going to have a nucleus. Some can have multiple nuclei, but most of them are just going to have one. So for a good example would be things like platelets or things like red blood cells. Mature red blood cells typically do not have any nuclei, but muscle cells can have hundreds, essentially. You also have this nucleolus here as well. Again, it doesn't really have a membrane itself because, again, just like the cell membrane is made of that lipid bilayer, the nucleus is as well. And again, this will dissolve in times of cellular division and whatnot, as we'll see later. But the nucleolus is where we're going to see where a lot of ribosomes and RNA is going to be found as well. And this can be very important for regulating things like gene transcription and new protein production. All right, then our ribosomes. Again, ribosomes we mentioned are important for what? Protein production, absolutely. That's one of the main things they're going to be used for. And it's important to understand that, like us as humans, we have a certain subset of ribosomes. You know, we'll talk about 70S versus 50S and all those different things. It's important that we have some very specific ribosomes that produce our proteins. But when you're dealing with things like, say, like invaders, like say for instance bacteria, do you think they have the same ribosomes? They actually do not. They have very different ribosomes, and that's what's important when we're talking about things like selectivity of different, say, like for instance, antibiotics. When we give an antibiotic, I want to kill off those bacteria, but I don't want to kill off the people, right? And so in those cases, I'm targeting specific types of ribosomes. So you're going to see this is a big target for antibiotics in bacteria. Here, though, and again, they perform the same function for bacteria as they do for us. We just have different varieties of them that code for different or allow for production of different proteins here. Now, there's several varieties of RNA that are going to be involved with protein production in ribosomes. We're going to find that messenger RNA, or this mRNA, is going to be the main thing that's going to be transmitting genetic information, say from the nucleus over into, say, like the rough endoplasmic reticulum, where you see a lot of the protein production happening there. So mRNA is very important for transmitting that information to the site for protein production. Okay, so any questions so far? Again, just trying to hit the main points here. Again, I'm sure most of you guys have covered this before. Just make sure we're kind of hitting it from kind of the medical sort of standpoint. Let's go ahead and take a 10-minute break. We'll come back and then continue on. All right, any other questions before I get back on this content bandwagon? So yeah, some people were asking me, they're like, some of this stuff seems really basic. Do we need to be like wasting a lot of time relearning a lot of this? No, if you inherently know some of this stuff, that's fine. The main point is to try to get everyone on the same page. Because did everyone take A&P like last semester? Some people may have, a lot of people did not, right? So maybe it's been some time. Maybe it's good to get a refresher. I like to put the medical spin on this because you may have learned it from somebody who is not a practicing clinician. I'm trying to put that spin on there. So I'm trying to give you like a lot of anecdotes and a lot of like little things like here's where we see this in the real world. That's the main point here. So if you can look at a monosaccharide and say, hey, that's a monosaccharide, I can obviously recognize that. That's fine. You don't spend a lot of time going over that. There'll be some sections here on the tail end of this where it'll be a little bit more intensive. We start getting into things like sensation. We start getting into things like the endocrine stuff. It's going to be a little bit more intensive, and that's where it's going to be a lot of the new material in regards to how do you translate this stuff over into an actual patient perspective. So again, don't stress over the basic stuff if you already know it that well. You don't have to go memorize things like the TCA cycle or any of that sort of thing. I'm not going to ask you to draw anything on the test, right? Again, I had a whole class in farm school where I had to draw structures and do all that. I'm not going to make you guys do that, right? I'm trying to get you the relevant stuff you need to know to be a good practicing PA. Anywho, all right, so going to the genetic control for protein production, and this is very important because we have all sorts of different things that can affect gene transcription and affect protein production, whether it be stress, whether it be infection, whether it be the medications we're administering, all sorts of different things can affect this process. So to know the basics is good to have in the back of your mind. Now, lots of different genes within a cell. We know that these genes will be coding for different proteins here. And the RNA is the important thing here. We're going to have things like RNA polymerase are going to be able to read that DNA and be able to produce a new strand of RNA from this, right? We mentioned the big difference between DNA and RNA. Yeah, uracil instead of thymine, again, usually only single-stranded you're going to see. So again, when you have that process of the DNA helix being unwound and having being separated out, this is where RNA polymerase can come along and start to make a new strand of RNA. This can then be moved outside of the nucleus, and then protein production can start here. And what do you call this process here? We're taking this information from the DNA, moving it to a new piece of RNA. Transcription, yeah. So we'll talk about translation a little bit later on, but transcription is this first part here, right? So imagine you guys are transcribing what I'm saying diligently. I'm sure everyone's taking down every single word, right? No, no one's doing that. Okay, that's fine. But you can imagine that would be a process of transcription. Same thing happening here. This information from DNA converting it over to RNA. Now, normally this code is going to be broken down to what we call these triplets or these codons, and so each of these three base pairs are going to be coding for what? One amino acid, right? So three of these will go into one amino acid, right? So again, UCU might go for, and again, I don't have that memorized, but maybe like a valine or arginine or something like that. It'll code for each individual one. Now, what happens if I were to say monkey with this and I change the C into like a G or something? What do you call that? Mutation, sure. Anyone know another name for that? You guys ever heard of a SNP? It's a single nucleotide polymorphism. That's another thing that's going to be huge in genetics. Dr. McClary, maybe teaching? I'm not sure if it'll be going with it. Anyway, but genetics is going to be very important with that because you can find that if you have a single nucleotide change here, you can drastically change the production of a protein, maybe change it to a whole new protein, maybe change it to something that doesn't function quite so well. It's a big deal in genetics, and we have a lot of genetic conditions that you'll be managing for these patients, especially because, you know, when you have a genetic mutation like that, guess what? It's a lifelong sort of disease you might be dealing with. Some of that lifetime may be pretty short if it's a very severe genetic condition, but some of them, you know, they may live with it for a long time. So it's important to kind of understand where these changes are happening. Another big thing that I deal with it from a pharmacist standpoint is this term called pharmacogenetics, which we'll talk about more in pharm later. But people respond differently to drugs, and some of it may be related back to these SNPs that occur when you have these mutations. But anyway, codons, they're going to each individually code for one particular amino acid. So that transcription is occurring from DNA to RNA. The RNA polymerase is the enzyme here that's going to be causing this process to occur. And again, there's lots of different enzymes that are involved in unwinding that DNA and causing those strands to break open so they can be read. I'm not going to get into detail on that right now. However, know that will be important when we talk about different therapies later on. But basically, it is going to know whether or not, or what sections to start transcribing based on things like promoters. They'll say, hey, this is the section of code I want you to transcribe. And then there'll also be these chain terminating sequences that say, hey, okay, we have all we need, and that's it. And then they can take that RNA, move it outside of the nucleus, and then start to produce new proteins in those ribosomes, right? So we'll have what we started with is called precursor mRNA, or this pre-mRNA. And so basically this is very immature. It's going to have a lot of excess genetic code it doesn't really need. That can be broken down into these exons and introns. Usually the introns are going to be bits of information that are going to be spliced out of the protein. Exons are going to be the stuff that stays in. So again, you have a pre-messenger RNA that gets chopped up, and again, different proteins will be, enzymes will be responsible for this, and then it'll get coded down into a nice tightly packed mRNA, and this is what's specifically going to code for that protein, right? So again, it's good to know the different types of RNA and what they're going to be used for, essentially, right? And so you can also have things like small nuclear RNA. These help to actually direct that splicing to make sure you get the right type of RNA that's actually being formed, the right sequence, and nothing extra is there, and nothing essential is taken away. All right, messenger RNAs are going to be carried out of the to the cytoplasm, and again, this controls the type of protein that's going to be formed. It's going to start to get involved here with the ribosomes. You're just going to kind of run through, and this is where we're going to have our transfer RNA actually come into here. And transfer RNA does what for us? It's going to be transferring amino acids onto this new protein, right? So you're going to see this transfer RNA. Notice it's going to have the complementary base pair. So if you have like a CCC, it's going to get pair up with a transfer RNA that has a GGG, right? And so these transfer RNAs are going to be associated with a specific amino acid. It's going to add that amino acid onto the chain, and then it's going to move off and go on to the next codon, right? And so by doing that, this is how we get this chain of amino acids. This is how we get our eventual protein being formed here. Now, do all these proteins come out fully mature? No, a lot of times they need some additional work, and so you have different enzymes that will be able to kind of chop them up, cut them up, and put them into different configurations. So just know this may be an immature protein, or it could be a fairly short one and maybe just good to go right off of there. But you find that there's also a type called a ribosomal RNA. These are going to be containing ribosomes along with other proteins that can be formed from this. And then there's some microRNA that helps to regulate transcription. And then what's this process when we're adding in the amino acids, or we're converting this RNA code into the actual protein? That's translation, right? So transcriptions from the DNA to producing the RNA to the actual proteins, that translation that's happening there, right? So again, microRNA itself doesn't code for proteins, but it can help to regulate this to some degree, okay? All right, any questions on that? Again, just the basics. We'll go into more detail where it becomes appropriate as we get into different systems later on. Anyway, cell cycles. You guys ever seen like Grease 2? They have that reproduction song. I'm not going to sing, but you can reference back to that. Again, they're essentially talking about mitosis. Wasn't anything else, just mitosis. I'm just kidding. Okay, so we have mitosis, which is what? How would you describe that? Yeah, cell replication, cell division, absolutely. At the end of mitosis, what do you have? Two cells. Are they different, identical to the original cell? Should be identical, right? They should be the same exact cell. They're just doubled off and separated, right? We'll talk about the individual pieces there. How about meiosis? What's the end result with that? You're going to get daughter cells. What about the son cells? You don't get any of those? No, we get short shrift there. Yeah, you're going to end up with daughter cells. And what are those daughter cells used for? Those are sex cells, right? So we're going to either produce eggs or we're going to produce sperm. The point here is that you notice that there's going to be a big difference here. Notice that the DNA is going to be changing here, because again, this is where we're going to end up having that shuffling of the DNA that occurs here. So that way you don't actually pop out as like a clone of your parents, but you're actually a mixture of the two. But you're going to have these sex cells that are going to be formed here. We'll go into more detail on this, but just know that this is what the ultimate production is going to be for these daughter cells, versus with mitosis, you're just getting two of the same identical cell, essentially. Okay, so cell division can be broken up into several phases here. So we're going to be talking about the G1 phase, S phase, G2, and then the actual mitosis that occurs here. There's also a G0 phase. Anyone know what G0 is? Yeah, cell's not actively replicating. We'll talk about that as being the quiescent phase, or kind of where the cell's just kind of trucking along, just kind of doing its job, essentially, right? But we'll talk about this interphase, mitosis, and then cytokinesis in more detail in a second here. But interphase, that's what ends up getting broken into G1, S, and G2. This will become very important when we talk about chemotherapy, and we talk about drugs used to treat cancer, because again, we said cancer is rapidly dividing cells, really without any sort of, any kind of regulation on those cells. Well, how do we stop that? Well, we got to stop them at the cell replication process. And so you're going to see this comes up again. We'll talk about things that can affect the cell cycle pretty significantly. And again, I already know your reviews are going to say he's only talking about farm, but trust me, you'll be glad to have heard it at least once or twice before you actually get to it later on. Anywho, so again, interphase, again, this is where you're going to see a lot of RNA protein synthesis actually occurring here. We mentioned G0 is where a lot of cells are just going to be kind of trucking along, doing their thing, not actively dividing. You're going to find that in some cases, there are certain cell types that they are only rapidly dividing. They spend very little time in G0. There's some cells that spend almost all their time in G0. So what would be an example of, say, like a very fast dividing cell in the body? Skin cells, absolutely. We need to be producing new skin all the time because we're constantly losing it, right? What else? GI tract, right? GI tract is going to be rapidly dividing because, again, you end up losing a lot of GI cells very quickly. What else? Hair follicles, absolutely. Anything else you can think of? Liver. The liver does replicate itself pretty well, but you don't see it's not as you don't shed a lot of liver cells typically, so you don't see the same thing like you would with the GI tract or the skin. Those. Yeah, so blood cells in general, right? So red blood cells, white blood cells, those are also things that are going to be replicating very quickly. Those cells can be very much affected by things like chemotherapy or with things like radiation. And so when you interrupt that, and when you think about someone who's on chemotherapy, what sort of side effects do you think about? Hair loss, right? So again, we've affected that rapidly dividing hair. What else? They have a lot of stomach pain. They develop what we call stomatitis because those mucosal cells are not going to be replicating because we've stopped that process. Yeah, deficient immune system, again, immunosuppressed essentially, right? Because we tamp down their ability for the white cells to replicate. So you're already starting to see, okay, well, yeah, those are rapidly dividing cells. If you interrupt that, you're going to be affecting them pretty significantly. Now, on the other hand, what are some slow dividing cells? They may spend a lot of their time in G0. Yeah, I like to think of neurons, right? And maybe not in the very young patients, but in adult patients, like where they're really not developing a whole lot of new neurons at that point. Typically, those cells spend a lot of time in G0. So you can kind of think of G0, cells just doing its job. G1, this is going to be kind of a growth phase. We're kind of getting prepared for subsequent phases here. You're seeing a lot of organelles starting to be copied because again, we're going to have to produce enough to make a brand new cell that's going to be fully functional. See, S phase is we're going to have the actual DNA replication occurring here. So again, the DNA is doubling itself. DNA polymerase is the main enzyme that's going to be responsible for this. Again, you should get 2 exact copies of that DNA at that point. And then the G2 is where you're going to start to see those chromosomes actually condense down. Normally in the nucleus, the genetic materials get much more loose, kind of free-flowing. Here, when it's getting ready for mitosis, you're going to see it starts to condense down into these chromosomes, which we're kind of used to seeing. Notice here that the copies are going to be bound by these centromeres here, right? So again, these are identical copies of the same chromosome, essentially, right? Remember, you have 2 copies of every chromosome, one from your mom and one from your dad, right? So again, I know this is basic, but trust me, you'll see where I'm going with this eventually. Maybe years down the road, maybe you've been working for 20 years, but eventually be like, oh yeah, that's what Dr. Wood was talking about. But it will happen at some point. So anyway, so again, the cells are, or the chromosomes are copying themselves. Again, they're going to be bound by the centromere here. Okay, and then we'll actually get into the actual mitosis phase, and I just remember the acronym PMAT: prophase, metaphase, anaphase, telophase. It's good to have at least some idea of what these different phases are going to be here. Now, we're going to see this is going to be similar when we get into meiosis as well, so kind of keep this the sequence in mind because this is going to come up again a little bit later. So typically, as I mentioned with mitosis, you're getting that cell division there. You're copying this DNA, right? And so you get a copy, you divide it out, and so each cell has the same identical DNA as the host cell did originally. That original cell, the same DNA. We call this diploid. What does that mean? Anyone know? It has both copies of each gene, right? So again, notice here's a red and there's a white, or each chromosome. There's a red and a white. Notice here, each of the 2 daughter cells has a red and a white, right? So they're diploid. They have both copies of all of their chromosomes, okay? Now, if it were only to have 1 copy, we're going to call that, anyone know? Haploid, right? So we're going to talk about haploid cells when we get into meiosis in a few minutes. But diploid means they still have both copies of all their chromosomes. They're fully functioning cells. They got everything they need in the world, right? Now it's prophase. Again, this is where you're going to see the chromosomes really start to become more visible as they start to get kind of tightly wound down into those the visible chromosomes. The nuclear membrane starts to disappear, right? So we're going to start to dissipate that, so that way it can kind of float a little bit more freely within the cell, because we need to get it set up for metaphase. And in metaphase is where we're going to have the chromosomes start to line up along the cell, right? And notice where they're going to be connected by to the end here. You want to know what these are? Yeah, so we're going to have these mitotic spindles, right? These spindle fibers are going to be bound to the chromosomes by the centromere, and this is what's going to get ready for them to separate essentially, right? So metaphase is where you're going to have that lining up actually occur. Anaphase, then the centromeres are then going to split as those spindle fibers start to pull it apart, right? So you start to see this pulling apart process to occur here, and then the cell is then going to get ready for telophase, where it's going to have the actual cytokinesis occur. So again, that the cell is actually dividing now. It buds off into 2 brand new cells, right? Again, genetically should be identical. Not to say there's not mutations that can occur, but they should be identical roughly to that host cell, right? And then you're going to eventually have the nucleus kind of reform again, and then the chromosomes can then kind of start to lengthen back out and kind of go back to the hopefully going to either G0 phase, or if they're a really rapidly dividing cell, maybe they're getting ready for mitosis again. When you have cell death, there's kind of 2 varieties that can occur here. You can have things like necrosis, which, how would you describe necrosis? You think necro, you think death, and basically the cells are dying off because they don't have enough of their nutrients they need, right? So again, if I starve the tissue of oxygen, your cells are going to undergo necrosis, right? Not good. You typically want to provide your cells with plenty of oxygen there. But it could be due to lack of blood supply. It could be due to injury. Lots of things like that can induce necrosis. On the flip side of that, you're going to have apoptosis, which is going to be that programmed cell death. This is going to be where we're actually telling the cell, hey, we don't need this cell anymore. Let's go ahead and get rid of it. Maybe it's damaged. Maybe it's not functioning properly. Maybe it's a cancer cell, a precancer cell we want to get rid of. Lots of different reasons for that. But again, it's much more of a kind of an organized sort of fashion versus necrosis, where cells are just kind of dying off pretty much unexpectedly from the body standpoint. Again, apoptosis is going to be regulated by a lot of different enzymes, things like caspases and things like that. It's important to know that certain cells, when they lack that ability to trigger apoptosis, that's when they tend to become cancerous, right? So we're going to find that a lot of cancers, they continue to rapidly divide and don't kill themselves off because they can't trigger off apoptosis. They end up losing one of those genes, or they have a mutation in it that makes it non-functional anymore. And again, we'll talk about that later. Okay, so when you're having rapid cell division, this is where you're going to have hyperplasia form. I mentioned hypertrophy before, and again, how would you describe the difference between these two?
Number versus size.
Yeah, number versus size. So hypertrophy would be an increase in size of the cells, right? Versus hyperplasia would be an increase in the number of the actual cells themselves, right? So again, important to kind of keep that distinction in mind because again, if you were to have something like a hypertrophic heart, again, muscle tends to not necessarily, when you have someone who, say, starts working out and they develop big muscles, it's not because they have brand new muscle cells. The muscle cells that they have to begin with got bigger for the most part, right? And so again, that's hypertrophy. Same thing can happen to the heart. Is having a big hypertrophic heart a good thing? Why not? It's a big muscle. It should be good, right? Yeah, chambers end up narrowing, they end up stiffening out. That's when you get things like CHF can develop from that, or congestive heart failure. So again, hypertrophic heart, not a good thing. When people tell you you've got a really big heart, man, they might be giving you a medical diagnosis. So just be careful with that, okay? All right. So when dealing with meiosis, you're going to see this ends up getting broken up into two phases, meiosis 1 and there's meiosis 2. Each of these are going to have their own prophase, metaphase, anaphase, telophase associated with them as well. But again, the difference here is what our end products are going to be. We're going to start out with a diploid cell that has both copies of all of its chromosomes. The end result should be 4 daughter cells that are going to be haploid, meaning they only have 1 copy of all of their chromosomes, right? Again, we should have 2 copies for everything for a normal functioning cell. However, these sex cells, so things like eggs and sperm, they should only have 1 copy. They're considered haploid at that point. So we're going to go over the detail on how this occurs here. All right, so everyone should have how many chromosomes? 23 pairs, right? So again, if you only had 23 chromosomes, you would be considered to be haploid. We have 23 pairs though, right? So again, you should have 46 chromosomes in total. What would happen, say, if I increase the number of chromosomes, say, like on chromosome 21? Yeah, so we get things like Down syndrome that can occur there, right? So again, lots of different things can occur here. If you're missing chromosomes, you have additional chromosomes, typically you want to have your 46, right? So if you ever get in an argument with somebody and they're like, well, how many chromosomes do you have? You don't want to respond back like, well, I got more than you, because it's usually not a good thing, right? It's usually kind of a problem. And also notice here, which one of these, I guess I didn't give it away on the top here, but how can you distinguish the male versus the female if I didn't label it? Yep, so look at the sex chromosomes right here. Notice you have this nice big X and a little tiny Y, right? So that's what makes those guys versus having 2 X's there for the female patients, right? And then obviously there's big change that can happen there if you have like an extra X or you're missing an X and things like that. So again, that's how you can tell the difference just looking at the chromosomes themselves. Okay, so meiosis, again, is this process. We're actually going to be getting these 4 haploid daughter cells that are going to be containing the genetic information you would combine with someone else's haploid cell to produce a new diploid cell that would be potentially a new person eventually, right? Now again, as far as the chromosomes go, we have homologous chromosome, which means that we have 23 pairs of chromosomes. Again, they each code for similar things, but they're genetically different and unique. So that way, they we have 1 copy for mom, 1 copy for dad. In some cases, you may find some things are going to be recessive in one and dominant in another. You may have similar copies of genes between parents. You may have different copies of genes. It just depends. But they're homologous because they're very similar in structure. Hopefully, we don't get the power to go out or something bad. But yes, 23 pairs are autosomes, and we're going to have 1 pair of sex chromosomes here, right? Meiosis, again, is that process. We're going to be developing eventually gametes like the ova and the sperm. Okay, so with meiosis 1, this is where we're going to have this reduction division that happens here. So again, basically, you're going to have all the chromosomes will double, right? So again, you're still going to have 2 copies of every chromosome developed there, so 46 times 2. But you're going to notice here during prophase 1, they have a crossing over process that occurs here. And what's happening during this crossing over? You're sharing DNA. So again, maybe a little bit of mom mixes with a little bit of dad, a little bit of dad. It's kind of like the Reese's thing. Like, you got chocolate in my peanut butter. Like, that's basically what's happening here, right? You're basically making a new Reese's cup, essentially. But we're shuffling the code a little bit, so that way we don't just produce like little clones of ourselves down the road, right? So again, my two daughters are very distinct from one another. One likes to sleep, and the other one absolutely does not, and was awake at 2 a.m. this morning until 7. So again, everyone's a little different. I'm not bitter about it at all, trust me. My wife, on the other hand, who's with them right now, she's a little bitter. But anyway, so then metaphases, we're going to have the cells lining up, or the gene chromosomes are lining up at random, and this is where they're kind of getting shuffled at this point. So again, crossing over, you can see a little bit of exchanging the genetic information here, a little bit of purple on the red, and again, that is good because we like genetic diversity, so that way we have good diversity going on, because again, if everyone showed up the same, that would not be very good for our genetic chances of survival as a species. Anyway, but then anaphases, we're going to see, again, very similar to what with mitosis, you have the chromosome being pulled apart, and then telophase, you're going to have them separate off, and so now you're going to have two haploid daughter cells. Okay, so this is the important thing here. So even though they still have the doubled up DNA right here, they're still considered haploid because they only have one copy of each chromosome, essentially, right? They have two copies of the same one, but they only have half of the genetic information they actually need. So this is the end of meiosis 1, is where I have two haploid cells, but they have double the genetic information they're going to have at the end of meiosis 2. Make sense? There's some pictures that kind of bit more clear in a few seconds. In meiosis 2, this is going to happen just like mitosis does, same PMAT format here, and then at the end, what you're going to develop are going to have these 4 haploid cells at this point, right? So I like this picture because it kind of helps me to visually memorize, helps me to visually remember kind of what the process is happening here. So imagine you're starting off with a normal cell. So notice you have 2 copies of every chromosome, and it's 2N stands for basically it's a diploid cell, right? So when it goes to 1N, that's when you know it's a haploid cell. Notice here when you have the S phase occur initially, and again, S phase is for what? DNA replication, right? So where all that DNA replication is happening here. Notice how you have 2C times 2, so now I got 4 copies of all my chromosomes. However, I still have the 2 basic groupings there, right? So I still have half from mom, half from dad, essentially. Once that prophase and the crossing over occurs here, they're going to get separated off. And then so now what you're going to notice is you're going to have 2 cells. They're going to each have a doubling of the chromosomes, but they're now haploid. They only have basically the 1 set from mom or the 1 set from dad. So again, there's some shuffling there, but that's how I remember it. At that point, you have another round of meiosis 2, and then this is where you end up developing the 4 haploid daughter cells, essentially, right? So again, they only have 1 copy of each chromosome, and then they can combine with someone else's and then produce a new diploid cell. Again, just another picture kind of showing the same thing. You start out with 46 chromosomes, you end up with 46 at the end of mitosis. With meiosis, you're going to end up with 23, again, 1 copy of each of those chromosomes at the end there. Any questions on that? All right, next off, moving on to talking about enzymes. What is an enzyme? It's a protein. It catalyzes a reaction, right? Perfect. What kind of reactions are going to catalyze? All sorts of reactions, right? We're going to find there's lots of different types of enzymes that are available to cause all sorts of different chemical reactions within our body, right? The main thing, though, is that that term catalyzes, right? Catalyzing being what? Speeding up the process, right? A lot of things can happen on their own, but it's very, very slow. We need to speed up the process, and enzymes is the way we're going to do that. So, for instance, you can have something like a substrate that will bind to an enzyme. The reaction will occur here and then do that enzymatic activity. Now you're going to have cleavage, and now you have 2 brand new products, right? So again, this is a very common thing you would see with like metabolism of substances in the liver. You have a lot of this cleavage that occurs here to get new byproducts, right? In some cases, you can have products, 2 separate products actually come together, and an enzyme will form a new single product at that point, right? So again, you can have catabolism, which anyone know what catabolism means? Breakdown, right, of things, right? So you can have catabolism, which is breaking down of things, or you can have anabolism, which would be building up of things, right? So again, enzyme can do all of this. But again, they're biological catalysts. They're going to increase that rate of reaction. They don't actually change the nature of the reaction itself. The reaction can happen on its own in a lot of cases, but just a lot slower. Notice here that a lot of these enzymes are not used up in this process. They can be utilized over and over and over again. They tend to be very efficient in that manner. So they can catalyze one reaction, go over, swim over, and catalyze some other reaction very quickly. To know they're identifying an enzyme, typically they end with an ASE. So you have things like acetylcholinesterase. So what do you think it interacts with? Acetylcholine, right? Absolutely. So carbonic anhydrase. probably acts as a carbonic acid, right? So we mentioned that earlier when we were talking about pH balance, right? There, amylase we mentioned is what? Sugars, right? It's going to break down sugars. We mentioned that's gets secreted along with our in the saliva. So again, if you see an ASE on the end there, you typically know it's going to be an enzyme. Now, these all have, enzymes can be pretty finicky in a lot of respects. So they need to be at a good temperature. They need to be at an appropriate pH. Some cases you can find the osmolarity of the blood can actually affect enzyme function. They tend to be pretty persnickety in that regard, but our bodies are designed to maintain homeostasis, right? So it's maintaining those conditions so that the enzymes work effectively. Again, they all tend to require energy to some degree. So this is where things like ATP can come into play here. But again, you're going to find they're going to have optimal ranges where they're actually going to be functioning most appropriately. Okay. And again, a lot of times with the enzymes, like the products themselves don't have enough energy to really get the reaction started, but the enzymes can kind of help to bring the additional, they kind of lower that barrier needed to get that reaction going. Okay, so again, notice that enzymes tend to be very specific, right? So again, think of a lock and key sort of mechanism here where only certain things are going to be able to interact with these enzymes based on what their function is, right? So for instance, say you have lactase. What do you think lactase does? Breaks down lactose, right? So again, very important because again, if you're lactose intolerant, what happens? No one ever dealt with anyone who's lactose intolerant? I have a, They can't break it down, so they get all stuck there in the GI tract, and they have horrible diarrhea, flatulence, very stinky. I have a cousin who likes to punish us during family events because he'll go and eat like a ton of ice cream, eat a ton of cheese, and he'd be like, sorry guys, and just chow it down. Now you can give people exogenous lactase, but he does not do that. So again, I know very well the problems of lactose intolerance. Anywho, but it's only designed to really interact with lactose, right? That we see in dairy products, right? So once it interacts with the enzyme, then it breaks it down into glucose and galactose, and then I can use that for energy production essentially. However, if I were to do sucrose, which would be two glucose molecules stuck together, it just doesn't fit. It doesn't really interact with that protein effectively. So again, lactose or lactase is not going to be able to interact with sucrose. It's only going to be specific for that lactose there, right? So enzyme specificity is a big thing. It's only going to interact with certain substrates as its form will dictate. So think of lock and key. Okay, so again, you're going to find that this is more of a kind of more abstract sort of picture, but if you ever see like an enzyme, you ever look at those like 3D models of enzymes, like all the ribbons and sheets and things like that, they're really cool looking, but again, they're kind of a lot of visual gobbledygook. So again, that's why I like to simplify to kind of a lock and key sort of thing here. But basically, enzymes can, either weaken bonds between different molecules to break them apart. They can strengthen that and bring two disparate molecules together to make a new product. It all just depends, right? So again, you can start off with substrate A and B, and then eventually end up with something brand new like product C and D, essentially, right? And some of these may be byproducts or trying to get rid of in the body. Some of these are going to be new active products that can be utilized for other purposes. It all just depends on the enzyme you're dealing with. So again, I mentioned that enzymes are going to enter that ASE, but there's lots of different varieties. So for instance, you can have things like phosphatase. What do you think a phosphatase would do? Yeah, take off a phosphate group essentially, right? You can have things like kinases that would actually add a phosphate group, and this is going to stay the same no matter what. So if you ever hear me talk about things like a tyrosine kinase, that's going to be an enzyme that's going to put a phosphate group onto something, right? If you see a dehydrogenase, it's removing hydrogen atoms. Hydrolases, isomerases, all these are going to be really important things. So anywho, we'll talk about these in more specific details later on, but just know that, as a class of enzymes, like they tend to have these names and they will dictate what the function of that enzyme is going to be essentially. Okay, so I mentioned this is influenced by temperature. So again, if the temperature is too high, proteins tend to do what? Denature, right? So think about an egg. You put it into boiling water. What happens to all those proteins? Normally, egg is nice and liquidy. You put it into boiling water, pops out. It's all solid now, right? Because all those proteins denatured, right? Same thing happens to you. You ever see that commercial? You guys are way too young for this, but that commercial where it's like, this is your brain as an egg, and they're like, this is your brain on drugs, and they threw it into the pan and all heated up. That basically can happen, right? So if you imagine someone who's like hopped up on phencyclidine or amphetamines, they get very, very hot, and they can fry a lot of those proteins essentially as they denature, right? So enzymes need to be within a comfortable temperature. You also need to find that they tend to slow down their reactions as they get lower in temperature. So we mentioned that therapeutic hypothermia. We can make people colder on purpose to try to slow down these metabolic reactions, right? So again, enzymes need that energy in order to function appropriately. pH can affect this pretty significantly. If you drop the pH too high or too low, that can also affect enzyme activity. A lot of times concentration of cofactors and coenzymes. We're going to talk about these in a minute here, but these are things that are necessary to allow the enzyme to actually function, right? These are going to be kind of supporting characters to help the enzyme do what it's supposed to do. And then also concentration of the enzyme in the substrate. So looking at, the enzyme itself, do I have too little? Do I have too much? That can affect things. And you can have people have enzyme deficiencies. So for instance, we mentioned acetylcholinesterase, right? I mentioned that interacts with what molecule? Acetylcholine, right? Everyone's heard of acetylcholine. It's really important for the parasympathetic nervous system, really important for neuromuscular function, important for lots of things. Acetylcholinesterase breaks down acetylcholine so that it can be recycled eventually, right? Because we don't want too much acetylcholine around. You can have people who are acetylcholinesterase deficient. They can have different mutations that actually make them produce less acetylcholinesterase. And then what do you think that does to the time of that reaction? It's going to slow it down because they don't have as much enzyme necessary to actually catabolize that reaction, right? The other thing could be the substrate as well. So if I had, say, too much of a substrate around, what do you think it's going to do to that process? Speed it up or slow it down? If I have a lot of product around, it's going to speed it up, right? Because again, all those enzymes can be used pretty very efficiently to get that process going. Again, if I have no substrate around, no reaction is going to happen. Okay. So, again, looking at temperature, again, an increase in temperature typically speeds up reactions. So you imagine someone has a very fast metabolism. What is their temperature usually like? If someone's like really ramped up metabolism, usually pretty hot or cold? Yeah, I think hot, right? So again, they have a very fast metabolism because again they are hot. They didn't generate a lot of heat. Those enzyme reactions happen a little bit faster. You kind of think about it like that. You get too cold, and they tend to slow down pretty significantly. Of course, again, with all things in moderation. If you get too hot, that's when you can have the denaturation. Again, a normal temperature for a person is like what? 98.6, right? So again, you can have patients that will get up into like, you think a fever like, 100, 101. You have patients that get like 106, 107, very, very hyperthermic. That's when you see that denaturation start to occur there. That can be very, very problematic. So again, a very narrow range we can really reside in, and it's important we stay within that range as best we can. Okay, and again, certain enzymes, this is the effect of pH on different enzymes. They work within a certain environment. So, for instance, pepsin. Pepsin is a very important enzyme that helps to break down what? Yeah. So it helps to pepsinogen eventually gets turned into pepsin, but again, breaks down proteins. Yeah, breaks down proteins. So pepsin is really important because it works best when it's in an acidic environment. So pepsin is going to work in the stomach, and what's the pH of the stomach? Pretty low, right? It's around 2 for most cases. Then that's important. So pepsin has been designed itself to work best when it's in a very acidic environment. If you move it up to a more basic environment, say it goes into the small intestine where things tend to get more alkaline, it's not going to work as well, right? So pepsin gets the most activity right there in the stomach versus something like salivary amylase, right? So again, the mouth, what do you think of its pH? Usually pretty acidic, pretty basic, or neutral? Probably neutral, right, for the most part. Well, that amylase, as it goes down into the stomach, as you swallow it, what do you think happens to its activity based on this graph? It's going to go down pretty significant. It's going to be almost nothing, right? You're probably actually going to digest that amylase as it gets into that acidic environment, right? So again, pepsin is made to live there. Amylase just really can't hang from that standpoint because the pH is too low for it, right? Trypsin would be another protein protease. They'll break down proteins. Again, it works best in the small intestine. It's going to be one that the pancreas actually secretes right there into the small intestine. So again, it needs to be, it's kind of like the 3 bears. You can't have be too cold, too hot. It's got to be just right for these enzymes to work effectively. All right, and again, big places you're going to see these large, like kind of wide differences, from the salivary glands to the stomach, back to the pancreas. You can find all these big swings in pH. It's important that we're giving the right enzymes to work in those environments. All right. Now, coenzymes are going to be basically, they're not the actual molecules that are causing a reaction to occur, but they're necessary. They need to be there to facilitate the actual enzyme doing its thing, right? A lot of times you're going to find that things like water-soluble vitamins tend to be cofactors or coenzymes needed to catabolize these or to activate these reactions. So things like vitamin B1 or thiamine is necessary for a lot of these, B2, B3, and you can find that when you have patients who are deficient in these water-soluble vitamins, they tend to not have these reactions occur quite so well, and they have diseases that can manifest because of that, right? So you have patients who have thiamine deficiency or niacin deficiencies. It can be problematic for them. I think would be cofactors. These are typically small ions and things like that that can help to cause the enzyme to work effectively. So, for instance, things like calcium, magnesium, copper, even zinc. Anyone know what you call these ions that are like in really small amounts in the body? There's like trace amounts of them. Trace elements, yeah. So again, trace elements are going to be ions that are in small amounts within the body. So think things like selenium, copper, manganese. You don't need a lot of it, but what you do need is there because of these enzymatic reactions. You need it to act as a cofactor for those to occur. Calcium, we have a ton of calcium in the body. We have a ton of magnesium, but a lot of these other ones is what we call trace elements, and that's where you're going to see those pop up. Okay. So again, cofactors are just going to help that process to occur. Again, the enzymes may be able to work on their own, but in a lot of cases, they work much better when they have the cofactors around to help to speed up these reactions. Okay, so again, looking at the substrate concentrations, right? So to a point, you're going to find that by adding substrate, by increasing the amount of the supply, essentially, the supply side of things, you're going to find that you typically will increase the speed of the reaction, right? So as I put more gas into my car, like I'm going to be able to make that engine run better, essentially. That kind of makes sense? So as I continue to increase the amount of substrate, you're going to find that the reaction is going to continue to increase. Now, at a point, can that continue forever? No, you're going to find it's going to kind of flatten out at a certain point. What do you think is happening right here? Saturation of the enzyme. You can actually have saturation of the enzymes where every single enzyme is busy doing its thing. Even though I'm increasing the amount of substrate that's available, the enzymes just can't work any faster, right? And so at that point, it becomes a very kind of flat curve. Doesn't matter how much more I increase the substrate, nothing more is really going to happen here. This is something that's actually interesting. I'll equate this back to not drugs necessarily, but to a drug, to alcohol, right? Again, none of you, I'm sure, ever have imbibed alcohol. Question whether some of you are even legal to drink alcohol, but regardless, alcohol itself, is very easily able to saturate the enzymes in the liver to metabolize it. And so we'll talk about this in Pharma a little bit later on this week. But essentially what you're going to find is it has this reaction where it doesn't matter how much extra alcohol you add on board, the liver can't metabolize it any faster, essentially, right? And so we actually use that clinically because I know that for most, say, non-alcoholic patients, I know that they're going to metabolize a certain amount of alcohol every hour. And I can actually say like, okay, about 20 milligrams per deciliter an hour, I know they're going to be able to metabolize that. And so what we use that for clinically is say I have like a drunk guy that shows up at the ER, so he got into a fight, got drunk, then got into a fight and shows up in our ER, and his alcohol level, anyone know what the legal limit for alcohol is? 0.08 or like 80 milligrams per deciliter would be the other units for that. So you can say 80 would be legally drunk. Well, I can't send someone home while they're still drunk, and so if I come back and their level's like 200, I can actually calculate when they're going to be sober, essentially, right? So I can calculate basically, well, okay, I know they're only going to do this much per hour. I know when they're basically going to become sober, and I can send them home potentially, right? That kind of makes sense? Or send them off to jail or wherever they're going to go if they got into a fight. Whatever the case may be. But we use those kind of things all the time in order to help us guide our clinical decision making, right? So again, just another clinical example to see how this stuff might come back. All right, and again, that saturation, this maximum rate is going to be achieved once you saturate all those enzymes. They just can't work any faster, no matter how much more substrate you put on top of that, right? Okay, so we can measure several enzymes within the blood itself. So these are just some examples of things that we can measure and some of the diseases that are associated with that. So for instance, I'm not going to go over every single one of these. However, things like amylase, amylase and lipase. Anyone know what lipase does based off the name? Breaks down lipids, yeah, absolutely. So it breaks down lipids. So those are two big enzymes that get released by the pancreas. And if you have someone who is complaining or has a clinical picture that looks like pancreatitis, those would be two of the enzymes you'd actually want to measure. You could see, hey, is our amylase and lipase really high? If it is, that may give you a sign that the pancreas is not functioning appropriately and you're having some issues with that, right? So you can diagnose diseases based off how these lab results come back. Again, you're going to get a lot more of that when you talk to Dr. Butler, next section, I bet. Things like, creatinine kinase or creatinine phosphokinase, a CPK, that can help us to determine things like myocardial infarction. What is myocardial infarction? It's a heart attack. What is it, though? It's ischemia of the heart. We're basically not delivering enough oxygen to the heart. What happens to those cells? They die off and they start to have necrosis, right? We mentioned that, and they necrosis, they just release all the contents out, and those enzymes can be measured. And so we are suspecting someone has an MI. I can measure something like that CKMB or that creatinine kinase myocardial fraction, and I can determine whether or not they're having a heart attack based off that. You can use other things, but this is one piece of the puzzle that can help with that, right? Transaminases are good for determining if you have like liver dysfunction that's happening there. We'll do a lot of monitoring for that, especially if you start medications that may harm the liver. But lots of different enzymes can be useful here. Right, and so going more into specifics of this, you can find that there's metabolic pathways, or there's enzymatic pathways that occur here, where basically you can start off with an initial substrate, product A, and it's going to go through several steps here to eventually get down to the final product, right? And so you're going to find, especially like metabolism of drugs, metabolism of a lot of byproducts in the body, you're going to find that it will go through these pathways where it's not just you metabolize it one time and it's done. It's going to go through several enzymes before it's really at a state where it can be, say, either peed out through the urine or go out through the biliary tract, wherever it happens to go. In some cases, you can find that there's branched pathways. So, for instance, these are distinct enzymes here that are working on different pathways, where you may end up with similar final products, but it's going to go down different trees here, right? And so, for instance, what would happen if I were to say I had someone who was missing this enzyme right here? What do you think would happen to this pathway? Say right now it's 50-50 between the two, but I take away that enzyme. So yeah, so this whole tree here is going to stop, right? So it's going to go down which branch? This one right here, right? So you end up getting shunted down one other pathway, right? I deal with this a lot with Tylenol poisoning, right? Where you actually end up seeing that you actually shut down one pathway and all the Tylenol gets shunted down a different one, and that's where they actually end up causing things like liver damage, right? So these pathways are very important to make sure they're all functioning appropriately, and then this is where you find a lot of genetic conditions and other metabolic conditions, where if you're missing an enzyme, you can end up shunting a process into a different direction, or you may shut down the whole thing entirely. So notice here as well that this is another example of a negative feedback loop that can occur here, where basically if you end up with too much of the final product, this can actually feed back and shut down some of these pathways. Okay, so if I end up having this tree being used a little too much, it can feed back, inhibit this process, and then shunt it up to this other branch here as well. Okay, so again, that can occur. Additionally, again, another example of a negative feedback loop. Now, has anyone heard of inborn errors of metabolism? You know what that is? These are basically genetic conditions where people are missing, or they may have a dysfunctional form of an enzyme. It could be one enzyme, it could be multiple enzymes. A lot of times you're going to find that when patients are missing some of these enzymes, it's not compatible with life. And you may find patients may not even be born because of these conditions are just too catastrophic to the body. In some cases, they may be living with these. And so working in a peds ER, sometimes we'd actually be the first people to diagnose some of these inborn errors of metabolism. A lot of these are caught early on after birth or potentially beforehand, but we'll go over a couple examples of what can happen here. So again, imagine you've had an abnormal gene. Say you had some mutation that occurred here, or maybe you had a defective copy from a parent. You're going to find that some of these pathways can be shut down, and then you end up developing, say, either too much of one product that could cause disease. That could be one pathway that could occur, or in some cases you may end up shutting off both of these, and nothing can be formed. Yes, ma'am.
Is PKU an example of this, or is that something else?
That's actually going to be one of the examples on one of the upcoming slides. Absolutely. Yep. So here's an example of amino acid metabolism, phenylketonuria, or PKU. It would be someone where they have an increase in this phenylpyruvic acid, and so you can see things like epilepsy can develop here, mental retardation, things like that. A lot of times, what I end up seeing a lot of cases are these diagnoses will end up developing epilepsy, right? And epilepsy is what? Well, basically the seizures, essentially. If you have a seizure disorder, you can call that epilepsy. And so that's a common thing that can happen there, where there's some defect that's occurring where they are more prone to have seizures. That's definitely one. Things like albinism. What is albinism? Some have claimed I have it. Yeah, basically they have a lack of melanin. That's where you have albino people come from, right? So again, they typically have very, very pale skin. They may have kind of red sort of retinas, as you'll see, due to that lack of melanin there. And then they can have things like susceptibility to skin cancer potentially, right? And being here in Florida, I would not want to be an albino person living here, right? Because again, a lot of sun exposure. And again, I always would be curious to see, what the you guys spend 8 hours a day in here. I would like to take like vitamin D levels of people like on this side of the room versus that side of the room to see, or maybe like skin cancer risk like in a 10-year period or something. See, I don't know. I'm just curious. They may have. You should probably wear sunscreen if you're over here though. Not today, but other days. Anyway, but you can have inborn errors of metabolism, lots of things, carbohydrates, lipids. Again, I'm not going to go over all these in detail. Just know that there's lots of errors that can occur here, and oftentimes they may either not be compatible with life, your patient's not going to last very long, or they can have very severe debilitating disease that can last a while, or it could just be things like increased risk for things like skin cancer. Okay, so this last little section here, before I wrap up, do I have enough time? like 2 minutes. Basically, we're going to get into energy production in the next section coming up. We're going to talk about ATP production and things like that. The basics here. Everyone familiar with the law of thermodynamics? What does it say? Yeah, so again, you can't make brand new energy. You can only have kind of these conversions here. So again, we're going to find that because we cannot create or destroy it, we can only convert it. And do you think we're 100% efficient in this process? No, we're not going to be. And again, there's going to always be inherent inefficiencies. Some people are more efficient than others, depending on disease states, depending on what conditions are happening here. But again, one of the big byproducts of this energy exchange is going to be what? Heat, right? So again, you have someone who has a very fast metabolism, they tend to be a lot hotter. You imagine someone who has hyperthyroidism, someone who has increased metabolism due to that, they tend to be pretty hot for the most part, right? Versus someone who's, say, hypothyroid, they can be more cold, right? Because they have a slower metabolism. But heat is a major byproduct of all of this. Again, all of our energy eventually comes from the sun at some point, and then through plants, they can develop things like glucose, and this is where we're getting a lot of our energy from. So again, you can see where kind of it all starts. The second law here is, again, we're going to lose energy with each transformation. So even though we're getting say a gram of glucose, we can't utilize all that energy that's possible there. There's always going to be a little bit that's lost in the transfer. And again, that available free energy is going to be that energy to do work overall is going to decrease through these processes. So again, you imagine starting off with a molecule of sugar and some oxygen here. Again, as you go through each step, we're going to be losing energy, we're going to be producing heat, and then we're going to get the kind of the end result product here. And again, all sugar, all these things are going, this aerobic metabolism ends up with carbon dioxide and water eventually. And that's important because carbon dioxide, we know, is a major byproduct. We need to get rid of that, usually through the lungs. Let's see. We usually refer to energy in intake, especially in our diet, as far as calories go. Where's my dietitian friend? She moved. Oh, all the way to the back? I scared you off that bad? Jeez. No. Anyway, calories are the main way we kind of show like kind of what the energy is going to be contained in a food, right? And again, the ability to get those calories out of the food may differ based on things like, the makeup of it, whether, it's fast-acting carbohydrates, slow-acting carbohydrates, with a lot of protein, different things like that can all affect it. But essentially, a calorie is going to be the amount of heat required to raise that temperature of a cubic centimeter of water 1 degree Celsius. Not super useful for us, but anyway, when we're talking about food, though, we're talking about it usually in kilocalories. But anyway, for whatever reason, maybe you know better than I do, we just call them calories, right? So basically, we call kilocalories a calorie. So when you look at something that says, hey, this has 150 calories per serving, that's 150 kilocalories actually. Okay, and then looking at this, we have to be able to exchange this energy, or this potential energy, into ATP. That's ultimately what we're going to be producing here. This is the energy currency that we're going to be utilizing in the majority of all of our metabolic byprocesses. And again, the mitochondria are going to be the main thing that's going to be helping us to generate ATP. Okay. We'll talk about the hydrogen molecule carriers a little bit later on. Again, I'm going to get into more detail on this in the next section. Just know that NAD, FAD, these are going to be things that are going to be able to carry hydrogen ions, and this is going to be a major source of where a lot of this ATP gets produced. And again, this is going to happen right in the mitochondria. I know I'm going over this real quick, but we're going to go to the next section coming up. So, any questions on section 2? I'm very happy I got through that. I was worried I'd be dragging behind this whole time. Yes, sir.
The diagnostic value of measuring enzymes in the blood, and like realistic conditions. Do we need to know the specifics on that?
Don't know the specifics. Just know that we are measuring them, and by measuring them, this can help us in our diagnosis, right? This can help us to determine how effective a treatment is. It can help us to find out lots of things. And so that's why we measure those enzymes, because it can tell us whether something's going on, something's normal, not normal, whatever happens to be. Any other questions I can answer? Awesome. All right, I will see you guys on Friday for our first farm class. Get ready for that. It'll be fun.